new test we are doing is to prevent cervix cancer. What is cervix? Cervix is, this is the uterus in which a child is born, and this part of the uterus below part is known as the cervix. Many women in India suffer from cervix cancer. So each year 5 lakh women worldwide get cervix cancer and about 2,70,000 die of this disease. And most of these are from developing countries. That is India, China, Africa. Because in developed countries, they have got very good programs to prevent it. So you have to remember that women between the ages of 30 to 69, 70 years are the most susceptible to this disease. And apart from dying from the disease, they suffer a lot because of the pain and the associated morbidity. So in India, this screening program is not there. Screening means even healthy people should go and get this test done. In India, what happens is, only when there is a problem, then the woman goes to the doctor, gynecologist, that time they do the pap smear. So what happens is, when they do the pap smear, we have done this study in our hospital that in 1 year, supposing 1,886 women have done this test, directly we find people with cancer. You see, 7 years, 43 people are directly found with cancer. If it was in any other country, you will not find cancer. Now what they do is, the other thing in which makes this cancer, which is associated with it is HPV, which is a virus. This virus this infection gets integrated into the cells and causes cancer. But there are more than 130 types of HPV viruses. Only 13 of these cause cancer. So now we have to do which infection is there and do the subtyping. So this is the method we do. You take the cells, isolate the DNA, do a test called PCR and do a gel run and know whether HPV is present or not and do a subtyping. But only few percentage of people who have this HPV infection get cancer. All of them don't get it. So which are these people? How do we identify? These people are the ones in which genetic changes take place. So there are chromosomes on chromosome 3, chromosome 5, chromosome 7 here and chromosome 20. These are the regions which have cancer genes, which are causing problems and developing a normal cervix into cancer. So this is the test we have developed. The test is known as fluorescent in situ hybridization is the method used. So what we do is, in the same slide, we put this particular probe which has been manufactured and validated between CGI and Kamineni, and we put it on the slide, do the washing and see it in the microscope. So in the microscope you will see different colors. For 3, if there is a problem, we will see changes in red dots. Otherwise 20 we see the yellow dots. Green is 5 and so on. So these are 4 different regions of the gene which help us identify it. So this is the pap smear and we count the cells. So if you see additional signals, we know it is abnormal. If it's normal, then there will be 2 of each. So what we have shown is that if you get normal cells or abnormal cells and you see whether HPV is present or the yellow color is the positivity with fish, you will see that as the cells go towards cancer, you will see more of the yellow, indicating that fish helps us identify genomic changes which cause cancer. So that is why what is recommended is that these 3 tests which is cell studies, HPV and fish need to be done as a triad, as a three test. And we have done a follow-up study for 4 years and found that if you do the fish, whenever you see fish positive, these are the cases which are progressing to cancer. So fish positivity is an indicator of cervical cancer progression irrespective of the cell test cytology, which we see earlier. So what we are recommending is that to detect this early, we should do the tests, and all women, they should not come to us after getting cancer. They should come when they are healthy so that we are able to do the screening. Now if a small thing of this, she will, Pavani Upendram, she has done this work and she has got the Young Scientist Award by the Indian Society of Human Genetics for this work.